Since only 8 of us, or plus me, I guess, are sociologists, I will give a one-slide overview of what the field is. So we're concerned with a bunch of things that I'll talk about today: race and ethnic relations, something that probably the most hot button when you talk about genetics. Inequality is a central concern of sociology, overlapping much with economics and other social sciences, but it's really core to sociology, and that involves understanding the distribution in a cross-section, but a very long tradition of looking at intergenerational mobility, which of course reflects both environmental and genetic influences, and why it's so exciting to be doing it in this day and age when we have genetic markers to incorporate in sociological studies. Also, another big area of sociology is demography and family studies, assortative mating, differential fertility, intrafamily investments, also overlap with economics. And finally, something I won't talk about today, I'll give examples of the first 3, but social cohesion, conflict, social change, networks and organizations. There's a lot of work that's just beginning that's trying to integrate genetics into network analysis. I'm looking at a couple of people I know in the audience who are doing it, and maybe they'll talk to you, during the breaks or at meals. I think it's really exciting to look at what some of us are calling social genetic effects, how your own genotype interacts with the genotypes of people, for example, in your class or in your grade that you are quasi-randomly assigned to, or in your family, which I'll talk about a little bit, or in how genes influence your network formation and propagation. So why should we care about it? Dan already mentioned it. We know, especially for things like intergenerational dynamics, that we're transmitting culture and economic investments in our kids and so forth, but we're also transmitting our genotypes, and that means that almost every model that a traditional sociologist has estimated for some intergenerational process has emitted variable bias. Of course, there's a number of solutions to that, but instead of trying to find the right natural experiment to isolate exactly the right environmental influence that you're trying to isolate, you can also try to control away as much as possible the elephant in the room of emitted variable bias, which is, of course, genotype. From a different perspective, and I've seen less work on this in sociology, the genome is a nice sort of log of sexual social interaction over long history, and I'll give some examples of that. We already talked about polygenicity. I'm talking fast because I know I have an abbreviated time slot. But I would say that for a field that was founded really 150 to 100 years ago in response to Galton, the trivia answer last night, and others, Herbert Spencer, who proposed social Darwinism and what called organicism, which is a biological interpretation of society in the late 19th century. Sociology was really a reaction to this sort of naturalization of inequality. So it's been very much anathema to sociologists to think about genetic effects. And the one area where it's kind of slightly socially acceptable is G by E. The notion that, okay, genetics might matter, but let's say there is a genotype for aggression. How it matters totally depends on social context. In fact, there's a crossover effect. So if you're, to the manner born, if you're really rich, like Mitt Romney, and you have the aggression genotype, you end up as CEO of Bain Capital. And if you're born in poverty in the inner city and you have the same exact genotype, it has a negative effect on your socioeconomic attainment. That's acceptable to say in sociology. So actually identifying, modeling G by E is, actually really hard, I think, because we already talked about all the issues about whether or not the genotype is random and how well we're measuring the genotype and everything. And then you have to add to that orthogonality of an environmental factor that you want to see how it works. And there's also other challenges because the way we estimate genotype is, as we talked about, an average across all these environments, the average treatment effect. However, identifying genotypes that are actual local treatment effects to particular environments is a different, it brings its own challenges. So I made it sound like you're basically doomed if you're trying to mix sociology and genetics, like, what is it, sodium and water, something makes the explosion. I forgot that from high school chemistry. But it's not as dire as I'm saying, and I want to say that I don't know if last year Dan had this kind of final day where he talked about sort of what you can do when you grow up with this, like careers in social genetics. I don't know if you're going to do that again. I won't be here, so I'll just say a brief sentence that, that it's not as bad as, I mean, I'm standing up here. I already have tenure, luckily. I've noticed a shift in the last few years. I mean, the fact that you're all here, there was many more sociologists that were not picked. There's a conference in Colorado every year called Integrating Genetics and Social Science that started with, I think it was like a dozen people 7 years ago and is now up to like 70. And I think actually in the better departments where there's more scientific attitude and less of an ideological or activist attitude, which permeates some sociology departments, there's an openness to realizing, you know, as Eric Turkheimer said in his famous law of behavioral genetics, I think it's the first law, right? All behavior is heritable. And in fact, there was a recent, by Jordan Smaller's group, a recent PHEWAS, a phenome-wide association study of UK Biobank, and the average heritability was 49% for every, I think they ran like 491 phenotypes in the UK Biobank, and so it really comes down to nature-nurture is like 50-50, 51-49. So we can't, I think sociologists are starting to realize that you can't really have a good account of human behavior if you're not in social outcomes, if you're not considering both. So I'll give you a few examples. Genetics, race, and ancestry. What does the genome tell us? Well, I think it's actually interesting to think about what we've learned from population geneticists so far, because it actually undermines it undermines both the narrative of the left and the right on race in sociology and beyond. So in terms of the undermining the idea of the left that race is entirely socially constructed, that is true. However, continental ancestry and population structure is real. There are real allelic frequency differences that have been a result of genetic drift, but also selection across human populations. And there's some evidence that selection pressures have sped up since the Neolithic Revolution. But I think it also supports some ideas of the left. For example, the idea of how we divide up race, at least, is completely a misfit for genetics. So if you look at this unrooted tree from a really well-known paper by Sarah Tishkoff and colleagues from 2009, and these are genetic differences in unrooted trees, so there's no like, it doesn't do common ancestry, it's just genetic distance. If you look at the distance between, say, Hasda and a Fulani tribe member, which are not, I think, about 400 miles apart in Africa, it's about the same distance as a East Asian to a European. So we think of these as equivalent races, you know, black, Asian, white, other in the United States, and it doesn't make any sense from a genetic point of view. We should not have St. Patrick's Day. We should have a bunch of African holidays and one for white people. So I think that, you know, this can be useful for understanding sociology of race and the claims that race is entirely socially constructed. At the same time, I mentioned that ancestry is real, content differences, population differences in alleles frequency. There's another paper came out around the same time, and basically on two principal components, and you're going to talk about principal components construction later, right? You can basically almost perfectly predict the ethnicities or the national origins of a pan-European, pan-Western European sample of respondents. So not knowing your self-reported nationality or so forth, by basically plotting you on the first two principal components of the data for a European sample, I can pretty accurately predict what country you're from. I think that's pretty amazing feat of molecular genetics. And these are the kinds of things we were talking about with the chopsticks problem of why you need to have controls for principal components, usually many more than two. Looking at in America, this is from AdHealth. You guys could reproduce this chart if you want later on this week. You can see that there are, again, challenging the idea of the one drop rule. There are people with African ancestry who have more European ancestry, people who are considered self-report as African American have more European ancestry than some people that self-report as white and so forth. And you can see patterns of admixture that, for example, Latinos, the green dots are an interesting spread of admixture across the first two principal components, with the first principal component, the x-axis, really reflecting African ancestry, and the second principal component reflecting Asian, what we'll call Asian ancestry, because the red dots are Asians down there. So you can start to unpack someone's self-reported ethnicity or race into something that's a quantitative variable and start fooling around with it, which we could talk about later. You can look at, this is from a paper in demography by Guang Guo at North Carolina and colleagues, looked at the specific forms of admixture. The most interesting group is to look at Latinos, thought of as one, well, not really one, but thought of as a sort of a racial group, but have very different patterns of admixture, as you can see. So, for example, Mexicans have a lot more Asian ancestry than other Latino groups in the United States. So, I'm going to other things you can do with these kind of data. Looked at identity by descent. It means common ancestral alleles. You're going to talk about IBD at all. Within siblings, it's used as a way to estimate heritability. However, across populations, you can see, for example, the degrees to which the thickness of the line indicates basically the average relatedness between individuals in different regions of the United States. And interestingly, you can see where there was interracial marriage, because this is within African Americans, this is within whites. So, for example, you see the southern whites have fewer connections to the rest of the country. They're more of a self-segregating, reproducing population. But if you look at this south central region, I guess it's called Texas and surrounding area, there's the highest degree of mixture between blacks and whites is between European Americans who come from that region and African Americans who come from the West. So, interesting things you can see in the genome that this is just another tool for social history that sociologists and historians could use that is now available to us that was not 10 years ago. So another thing you could do is Dan mentioned using genes basically either as control variables or as instruments for what's called Mendelian randomization, something I don't believe in at all, which we can talk about later. However, you can do other things. You can start examining the social effects of race, for example. So this is in, again, AdHealth data. So you could go do this. Looking at skin tone. They have a measure of skin tone in AdHealth, and this is on diagnosis of hypertension. And if you look at everybody, there's a slight effect. Sorry for no error bars, but these are significant. And if you look at understandably, the effect is coming from African Americans. So here we're comparing siblings who differ on skin tone, and we're finding that the darker skinned African American sibling has a higher likelihood of being diagnosed with hypertension. There's two potential threats to this as a causal interpretation. As far as I know, there's probably more that I haven't thought of. But one is that behavioral differences result in skin tone differences. So let's say one sibling's a ditch digger and has a stressful outdoor labor life, gets more sun exposure, is darker, but it's the labor, the hard outdoor life that's causing the hypertension. We can control for that. We did control for that. The second would be genetic pleiotropy. So whatever the effect of the genes that determine skin tone are also having an effect on the cardiovascular system. So this actually turns out to be only 8 SNPs that you can characterize eye color and skin color with, and you now, because we have genetic data, you can control for those in such a model. Yeah.
So earlier we were talking about how, at least in sociology, there's like a lot of taboos surrounding using genetic data. And I guess so one question I have is, how do you frame, like, it seems like we're fitting things into a very particular potential outcomes framework. And like, so in this case it would be like, all right, if I had different genes at birth that caused me to have different skin tone or different predisposition to skin tone based on certain environmental exposures, then we're looking at the causal effect later, and that's the causal effect of skin tone on hypertension diagnosis. But like, I feel like a lot of sociologists or other social scientists wouldn't want to characterize that as the causal effect of skin tone. It's like the effect of, you know, a racist society or something like that. Like, I guess, how do you negotiate sort of that difference, you know?
Well, yeah, that's, I mean, the hypothesis is that it's working entirely through the social response to the genetic endowment in the form of skin color. That it's showing the social effects of discrimination. But that's an extreme example of everything we're talking about that work through RGE, or evocative RGE to be specific. In other words, I don't know what proportion has been estimated as of late in your guys' analysis for EA3 or something, but something interesting to think about is the fact that much for educational attainment, for example, probably more so than for, say, something like height, but in modern society, is working through evoked environments. It's the genes that cause the kid to tug on the parent's sleeve and ask for one more book before bed that leads to the higher verbal score. And in the potential framework, potential outcomes framework, then, you know, absent an environment that allowed that parent to respond to that genotypically induced request for an additional book, that there was no books, that there was the parent had some disability, whatever it is, then you wouldn't see that effect. It's contingent on the environment. It may work through the evoked environment, and that's part of the genetic effect. Is that what you were going to say, David?
Yeah, so basically the issue you're raising is exactly the Sandy Jenks critique that we'll talk about tomorrow when we discuss heritability estimates and how to interpret them. And Jenks's point was just that, you know, whenever you observe one of these genetic effects, he actually used exactly the same example that you just used. It could be that I have some physiological character response and that ultimately that affected my earnings or something complicated like that. That's still a causal effect, and people who are skeptical of the behavioral genetics enterprise ought to spend more energy focusing on the interpretation of the estimates rather than the estimates themselves. And it's absolutely true that the way we've defined a genetic effect, it would capture, it could be incredibly complicated causal pathway that operates through all kinds of environmental mediators. And yeah, we've thought about this a lot, and kind of we think that's the most coherent way to go about it. But it's a very important interpretational caveat.
I think this field is very young, and so because of power issues that Dan will be returning to again and again, we've really focused on trying to get the average additive effect for most work, and then I think, at least in my head, down the road, is having enough power to look at the heterogeneity effect of effects in response to different environmental constraints or availabilities and so forth, and treatments. So I think that's a longer term payoff that many of us are hoping to get to. I'm going to skip through this, but this is an example of from Ecuador, where there was very few written records during the encomienda period about mating between three populations: the indigenous population, the African slave population, and the conquistador population. And yes, some things were obvious, like the conquistadors were men, raped women. You see a disproportionate amount of European ancestry on the Y chromosome, but some things were not known until we had this essentially historical record that, for example, there was a lot of seemingly bilateral, in terms of gender, exchange of DNA between the African population and the native population. For example, that there, in fact, I think there was it's the most admix between native and African populations. It's the country with the most amount of admixture between those two groups of any country in the New World. So this is something that we couldn't learn from traditional history, but we can learn from DNA. I'm going to skip ahead to stratification, unless there's any questions there. Yeah.
So I'm just curious, is the genetic data sets available racially skewed? Because my impression based on the previous reading is that a lot of them are based on European ancestry, but we're curious about racial inequality.
Did everyone hear the question? Are the data sets available racially skewed? And there's two answers. The answer is yes to both. So for almost everything you're going to hear about today, it's been estimated predominantly or exclusively on those of European descent, non-Hispanic whites in the census terminology, and you'll talk about different linkage structures by population, and that's really the reason. In other words, a allele that predicts educational attainment in one group, those alleles are not the actual, in vast majority of cases, are not the actual causal alleles. They're just kind of flags planted in the chromosome meant to cover an area that, because of linkage, travels together. So the actual causal allele that's affecting educational attainment through either brain development or evoked environment or whatever it is not what we actually measure. It's something that is traveling along with what we measure to a certain degree. And because there's such a great wellspring of genetic diversity in sub-Saharan African populations, the rest of us were descended from the same approximately 1,200 people who made it out of Africa. There was incredible population bottleneck that reduced the amount of genetic variation. So as a result, when we have that SNP number RS1234 on chromosome 1 for a European or Asian population, that's tagging a much bigger part of the genome than it is for a sub-Saharan population. So, for example, there was a nice example given recently that let's take a non-controversial phenotype like height. Taking a polygenic score trained on Europeans for height and then trying to predict height in a black population, it predicts the mean to be 6 inches below what it should be. So it just simply does not predict across populations. There are other things you can do to try to make inferences across populations, but it becomes a very tenuous, that's a long discussion itself. But yes, there's bias. There's also bias in the 1,000 Genomes Project about which is kind of the reference panels to impute to what populations were sampled. In fact, there's a nice map that can show like what was actually sampled and what the world would look like if that was the world. I'll try to dig it up, versus what the distribution of the population actually is in the world.
And just to point out the obvious, so it's very likely true that as a result of this bias, in the short run, any medical gains coming from the new GWAS discoveries might disproportionately benefit European ancestry individuals. That's a real risk that people talk about a lot, because that's where you have the largest discovery samples.
Yeah, I mean, there's a whole bunch of reasons for that. One of them is just level of development of the countries that pay for these studies. Yeah.
I've been mulling this over in my head for a long time, and I mean, assume we have perfect genetic data on everyone in the world, on every ethnicity, and so we can perfectly measure like a polygenic score in every population and things like that. I mean, would we really find differential effects of, say, the social environment? Would we really find a different G/E estimator? I mean, because even though we can't measure these polygenic scores perfectly in these other populations because of the LD structure, but the functional gene is still the same. It's just that the LD is different on that gene. And so, I don't know, I guess to the extent that having more data, it's important and we need to do it. I'm not saying we don't need to do it. But to what extent will that really change the findings that we're, say, finding in European Americans? It could matter on like a very small genetic level, like if you're really like one functional gene for like a medical intervention, or like a particular SNP or something. But is it,
I think, I'm not sure I understand the question, but there's, as long as you're using genotyping arrays, then you're stuck with a problem with like if your thought experiment is that we actually fully sequenced everybody in the world, I think you could develop, and there was, an infinite number of people of all different descents, you could solve the problem. You still couldn't make comparisons across, like you couldn't use the polygenic score for African Americans and whites and say, I'm going to explain the test score gap, because there in fact are what are called private alleles that you only see in one population and not the other. And of course, like I said before, the most private alleles are in the African descent populations because there was greater well of diversity that was not eliminated. So you would have different scores estimate. So I would imagine that if you had this thought experiment and you had full genome and you estimated within those groups the effects of the allele and developed polygenic scores that predicted as optimally for European Americans and optimally for those of African descent, then you wouldn't be able to use it to, for example, just as a control. If you used one as a control variable to see if, well, could you explain the black-white test score gap, it wouldn't work because probably the African one won't predict in Europeans and vice versa. I mean, I haven't thought through the thought experiment entirely because we're way away from that.
I guess my question is to what extent, I mean, because the LD structure matters, but the functional gene is still having the same.
Well, sure, but most of what is driving phenotypic variation is not structural variance in genes. It's regulatory regions. So I'm not sure the fact that we all have the same genes solves the problem. Yeah.
So I know that you said that most of the work has been in white non-Hispanic populations. Does that mean literally that there's not data for other populations, or just that work has not been done?
There are some data, however, there's less data. So there is Fragile Families, run by my colleague Sarah McClanahan, is a predominantly Latino and African American population. It's a small sample. In Ad Health, there's, as I showed you, there's non-whites. HRS has they all have, but they're not samples meant to maximize minority representation. They're nationally representative samples. They might have, like PSID has an oversample of African Americans, but not enough to really, we don't have enough to do discovery, really. And second, because with respect to the African American group in particular, because of what I said, that there's greater diversity in the genome of those of African descent, you actually means you need a larger sample size to train the scores on. So not only is there a smaller, but in relation to what you need to develop a good score, it's way less. I don't know the exact figure, but you would need a much bigger sample because each tag SNP is picking up less of the genome.
Sorry, just quickly, but going off that, is it true that genetic diversity of African Americans is as great as it is for Africans?
It rivals it. Yes, I mean, even though there's an admixture of like approximately 10 to 14% European descent. In the same article I just showed you with the unrooted tree, Sarah Tishkoff, you can look in this supplementary material, and she plots measures of diversity, heterozygosity, against, I forgot if it's copy number variance or something like that. And she plots all these different groups. And you look closely at the tribes. It's like Fulani, this, Hasda. And then you see Pittsburgh and North Carolina. I was like, what's that? And it turns out she had samples of African Americans in the US. And they're right there. They're not the most diverse, but they're right in the thick of things in terms of the African populations. And there is, as many of you might know, I'm looking at Omer, diversity in the genome, that was the big bottleneck, but there's lots of other bottlenecks. And it generally goes down as you, in sort of distance traveling by foot from East Africa. Yeah.
Would you agree with the statement that another challenge that we have working with minority populations is, and I think he alluded to this, is the fact that not only they can have unique genotypes, but also they're exposed to unique environments. So for example, I did my dissertation without health data. I wanted to look at like cultural proxies like acculturation, which only found like languages at home. And for racial discrimination, there's one item in wave 3 that is like, are you discriminated? But to make things more complicated, what happens sometimes is the way that so many groups experience these effects are unique, and in psychology we use measures that are targeted to these groups. And at the same time, they live in unique neighborhoods. So an ideal project would have not only the opportunity to genotype them, but also deep measures that assess their unique environmental experience. So I think that's an added complexity that at least we're dealing with now that we're trying to genotype and measure these groups.
That's absolutely true. I mean, earlier, Guangguo published a paper arguing that there was environmental repression of African American genetic effects on cognitive development and education. This was based on twin data, not measured molecular data, but we still, and the argument was that, you know, with a disadvantageous environment, a lack of economic resources to invest in kids and so forth, social stress, et cetera, the genetic differences inherent in the population aren't allowed to flower. So you actually see reduced heritability of IQ for African Americans. And this is similar to what Eric Turkheimer claimed for by socioeconomic status. However, it becomes a difficult problem to unpack because there is the different allele frequencies and haplotype structure as well as a different environment. So that's kind of one of the challenges to try to say, to identify G by E effects, particularly across race.
I just wanted to mention, NIH is prioritizing data collection on non-European populations. So I think over the next few years, there's going to be a lot more data on non-Europeans.
Well, I don't have a lot of, I'm only like 1/3 through my slides, but okay, I'll just say, you know, the problem of unobserved heterogeneity is a big deal. It came to me in the form of this book 20 years ago. Susan Mayer, who's at University of Chicago Harris School, where she showed that the effect of family income is vastly overstated in traditional regression models using a number of tricks. And yes, there's been like the Sacerdote papers on the Native American windfalls, and there's been David's work on Swedish lotteries, and the effect of income on kids is not nothing, but I feel like this book like reset the agenda, and we had to rebuild our understanding of the causal effects of income. And finally now, there's two basic confounders: cultural transmission that's associated with labor market success, but also with kids' health and well-being, and genetic transmission of alleles that affect parents' labor market success, i.e. income, and then also the kids, if we want to actually properly estimate the effects of transfers, let's say. So then there's also, around the same time in the 90s, the bell curve. I won't even get into this.
John, it's okay. David, you can take more time if you...
Yeah.
Okay. So they use the NLSY 79 with a very narrow window of birth cohorts from 57 to 64, yet even within a 7-year window made this sweeping historical argument that the returns on IQ have increased dramatically in the 20th century, but they had cognitive ability that's obviously endogenous to environment because they were measuring the actual test scores in the NLSY, and like I said, they had a narrow birth band. So I thought it would be a nice uncontroversial thing to do, to go back and evaluate their hypotheses using molecular data that we now have. So you're all going to talk about this, but basically there are two types of variation that are commonly measured. One is a single nucleotide polymorphism. That's a single base. If you remember the trivia game last night, ACTG. And the other are copy number variants. Those can be large things like what are called indels. They can be microsatellites in this case, where there's just a single motif repeated different numbers of times in different individuals. What we're focusing on is I know that in some of the analysis you guys have done, you're looking at structural variants, but mostly we're focusing on single nucleotide polymorphisms in our construction of polygenic scores that you're going to spend a lot of time talking about. I've already talked about this. So we talked about the polygenic score, which tries to pick up this additive component by simply summing across, depending on how you do it, maybe independently sorting SNPs, maybe not independently sorting SNPs. There's lots of analysis. Lauren's done a bunch in HRS to see how much you should prune for linkage disequilibrium or not. Basically, since we're basically interested in prediction, whatever works in terms of how many SNPs to include in your score. I'm sure you guys are going to discuss all of this, right? And what's nice about it is you get a single scalar that, like in the movie Gattaca, if you haven't seen it and you're in this field, you should go watch it on Netflix. You can predict somebody, from birth essentially, or even pre-birth, predict someone's height, for example. These are even old. I stole this table from Dan, and it's not even been updated, but the R-squared for height, which is the best current score, I think is now in the 20s. Is that right? Anyway, you can do, and for educational attainment, the last paper was 7%, but now it's double digits, I think, right? You can, again, this is averaging all Bs across a population, and since these are derived from white non-Hispanic samples, but across from David's social democratic Sweden to US contemporary ad health, kind of Yankee capitalism, across all sorts of environments that social scientists and policymakers are typically interested in. So this is an average effect across all of those. So something you might want to keep in mind as you think about gene by environment interactions these might be the most robust genetic effects across environments. But the point is that we have this score. You could prick your kid's heel, like in Gattaca, predict their height, a bunch of error, educational attainment with a bunch of error. I decided to go crazy again.
Life of its own.
There's a question? Yeah.
You said earlier that a lot of this stuff is around 50%, but everything on that list except for height was lower. So are somehow these things that we're so interested in all less heritable than the typical thing in some sense?
So this is SNP chip-based heritability, which is generally lower than even full additive heritability because the SNPs on the chips incompletely tag the causal alleles.
But I mean, even the H squared column.
That's what I'm talking about.
Oh.
Yeah. So although I think that paper that actually is also doing SNP chip-based heritability and got 49%. So that's a good question. Yeah, height is definitely higher than this now. So I think these are old. And anyway, this SNP, so there's broad sense heritability, which includes dominance and epistasis and everything. There's narrow sense additive heritability, which includes all additive effects, even on all the alleles we're not measuring, and rare variants and so forth. This is the variation due to the common variants that we're measuring on a typical chip.
But at the conference a while back, they were saying what we could theoretically get from the chips with the additive model was getting pretty close to what you get from twin studies. So I guess the question is, do many of the things that social scientists are interested in, are they just below that sort of 50% average, even when you look at the twin studies, which presumably include most of this stuff? To what extent is a gap of the methods, or to what extent is it that the things we're interested in are especially low in heritability?
That's a good question. You want to?
I hope you're not referring to anything I said, because I don't think that we are, that we're about to get close to the twin-based estimates of heritability. Yeah, I think it is realistic to expect in the coming years, and we'll look at this theoretically later on in the week, I think, or maybe next week, that we'll start to get fairly close to the H squared SNP column in terms of predicted power. But the twin estimates themselves tend to be, a bit higher. In the case of height, maybe 80%, and that's going to take longer. You're going to need sequencing data and all these rare variants. You're going to need very, very large samples to get precise estimates of the effects of rare variants, because there's more estimation error the lower the algorithm frequency.
And there's also a debate about whether you can get, using the classic twin model, which you'll talk about tomorrow, you're getting broad sense or narrow sense heritability, because with the identical twins, you're getting all of the effect of epistasis, because they have the exact clone. You're getting the dominance and epistatic effects, and with the fraternal twins, you're not picking that up. Oh, you're picking up to 1/4 of that, even though you're actually, if I'm remembering correctly, you're treating them as half genetically similar, but for let's say a biallelic epistatic interaction, they're actually only 1/4 similar. So on average, don't quote me on that, but that's basically you're getting underestimate of the it's including part of the epistatic and dominance effect in the classic twin model. So I'm not sure we'll ever get to that. And then there's issues about prenatal architecture, prenatal environment. There's a bunch of other issues that come up.
On a specific issue of cross phenotype differences, the Polderman et al. Paper, which is in the reading list and in the folder, actually looks at heritability in twin studies across different types of phenotypes, and it is the case that there's a lot of medical phenotypes, like directly things like circulating lipid levels that are highly much higher heritability than the kind of behavioral phenotypes we look at. And I don't know this UK Biobank paper that Dalton mentioned, but the UK Biobank has a lot of health measures, so it wouldn't surprise me if they're if they have an awful lot of those kind of medical phenotypes with very high heritability. In general, the behavioral phenotypes, the twin estimates are almost all lower than 50%.
Like the twin estimate for educational attainment, just years of schooling, I think is around 40% in the meta-analysis. And then chip-based is about 25%, 20 to 25%, right? So that's what we're talking about. Subjective well-being much lower, but other behavioral ones probably higher.
Oh, quick question. Are these results all from meta-analysis or from individual cohorts?
These are all from meta-analysis as far as I know. That's how they have these big sample sizes. I mean, the only two single biggest studies are UK Biobank, about to be released the full data, 500,000, and then 23andMe, which is over 1 million. I don't know how many you can actually get, but is a very skewed sample. I mean, they're all skewed for different reasons, but that's probably the most skewed of all, since it's all people like us who send in our saliva. Yeah. Okay, so this is getting to a question about whether or not control for principal components actually deals with, is an adequate substitute for the within family model that we've been talking about as the ideal experiment. And you can see when we looked, this is the effect of the first score for educational attainment that was computed in 2013 with no controls for PCs. This is, again, remember, this is restricted to whites, non-Hispanics anyway, but as we talked about, there's lots of substructure within the population. This is controlling for 20 PCs, and this is within family. So you see that it's pretty consistent. So if we control for at least 10 PCs, let's say, you're approximating the idea of the randomness of the alleles from the parents' transmission. I'll come back to that issue. Normally distributed, thank you, Lauren, for these slides. That's HRS. So for example, currently, this is with was this the first score or the second score?
This is the
first score, I think, right?
This is the first score.
So this is out of date, but if you're in the first decile, you're predicted to have basically no more than high school, and if you're in the 10th decile, you're predicted to have at least an associate's degree. So it predicts pretty linearly, as you can see. So once we have this tool, we can start to evaluate a bunch of hypotheses. So for example, I took a bunch of quotes from the bell curve that the effects of genotype are increasing over the 20th century and the effects of environment are decreasing. You can see this in a bunch of phenotypes. For example, there's a polygenic score for cigarette smoking, and you can see that it significantly gets more predictive over the course of the 20th century, or at least birth cohorts 1920 to 1960, so that, which we understand that early in the 20th century, the norms about smoking were completely different. The understanding of the risks of smoking were completely different. It was much more common to smoke, and then leading up to and post Surgeon General's report, there's a dwindling number of smokers, and those who smoke today tend to be those who are genetically most wired to like nicotine, is the hypothesis, and we can see that the penetrance of the score increases over time. There's a number of estimation questions about this. For example, you could just get this artifactually if your discovery sample predominantly came from younger age groups, and therefore your score was trained on people born in 1960, and therefore it's not that genetics mattered less in 1920, but maybe different genes there was a different predictive score that mattered. So you need to be aware of all these kind of things when you're doing an analysis like this. It turns out actually, if anything, we're biased downward because most of the discovery samples, I think the mean was in around 1930 something in terms of discovery samples. So if anything, it was biasing us to find a better effect, a bigger effect in earlier birth cohorts. There's, of course, especially with smoking, is the single biggest one where there's mortality bias. People have to survive to be genotyped, and smoking kills you pretty quickly. So that's the one that shows the biggest mortality bias. If we look at height and BMI, is that a question? No. If you look at BMI, so this is someone who is 1 standard deviation below the mean on their polygenic score for body mass index, and this is someone who is 1 standard deviation above the mean in their polygenic score. As you can see, everybody is getting heavier across birth cohorts, but they are diverging. This is statistically significant. So, and again, gene-environment interaction over the course of the 20th century, as obesity rates rose and caloric environment changed, and the amount of physical activity we do change, it allowed 1,000 genomes to bloom. So you see a gene-environment interaction over historical time, such that not only is the mean and the variance increasing, the genetic penetrance for BMI has increased over time. So you could ask, well, what about education? And we find a slight decrease in the effect of the educational polygenic score on predicted years of schooling in the United States. In the appendix of the Educational Attainment 2 paper in Nature, this same sort of analysis was done for Sweden, a very different social context, and found the same effects where there was just actually, I think, surprised everybody that the genetic effects for educational attainment were decreasing over the course of the 20th century, not increasing. But again, you could imagine a story where if you were going to go to college in 1920, and particularly for women, you probably were genetically really endowed, and you kind of fought your way to go to college. And when you make opportunity available to all, like, it doesn't necessarily follow the logic of, say, calories for all, where genetic differences flourish. It can actually reduce or attenuate the effect of genetic differences. So we don't find support that for the bell curve claim that, yes, education is definitely partly predicted by genotype, but that doesn't seem to be accelerating, or genotype is not becoming more salient now in some very obvious way. Yeah.
So maybe this is a question I should save for later when we talk about the polygenic score, but it just seems to, I can't stop thinking about that the polygenic score is based on people that lived in a certain time and lived in certain countries where they would have a certain access to education. So if I go to another society where the access to education is very limited, I might not find a polygenic score that would be the same, right? Or
that's exactly right. Yeah. So we're taking all of these claims are tenuous or tentative based on or applicable only to the environments under which the score was trained. So it's sort of modern Western countries in the 20th century birth cohorts. So it could be, for example, that in a different context, the genes that predict the endophenotype of assertiveness and aggression actually predict educational attainment much better in conditions of economic scarcity or educational scarcity, because you have to be very assertive to be able to go to school. All genetic, something you should keep in your head front and center all the time is all these genetic effects are, there's nothing that's like universal biological. They're all contingent on the social environment in which they're estimated. So you could even imagine something like so biological as BRCA2, the breast cancer gene. So Angelina Jolie gets double mastectomy. So right now, BRCA2, the risk allele predicts 8-fold risk of breast cancer as compared to the safe allele. You could imagine that completely flips because of social behavior in which people preemptively get mastectomy. So what BRCA2 predicts is preventative mastectomy, and actually lower, because of that, 0 breast cancer risk, and people with the safe allele are only ones left to get breast cancer. I mean, so the system is changing and learning all the time, even from the information, the genetic information we provide. So it's all historically specific. Yes.
Do you have a take why we see that? Like, that doesn't leave this room sort of just like throwing out a couple of...
I'm on video. We're number 1 for the web, so everything's going to leave this room. But go ahead and ask.
For like, just the thing you were talking about in Sweden and in like that US cohort of the decreasing penetrance of the education score, like, do you just have like an a priori, like, oh, this might be happening?
You know, I think that we could construct 2 competing stories that would have explained whatever let's say we found the opposite. You would have said, oh yeah, well, repression of genetic effects, that there was very limited opportunity for education. It was kind of random. George W. Bush went to Yale because he was child of an alum and a president or whatever. Like, you know, there wasn't sorting on the education system based on anything meritocratic. Therefore, we should expect it to increase now, like Herrnstein and Murray expected. But, you know, we didn't find that, and you could say, well, actually, compulsory schooling, all sorts of things. Social norms have changed, that maybe people who, quote unquote, you know, don't benefit as much from college go anyway. There's probably much more horizontal stratification within the education system today than there was 50 years ago. I don't know. It's all speculation, just so stories, you know, but it's something worth understanding. So another question is whether or not we're sorting more genetically, as they claimed. You can do a genome-wide assortative mating measure, which is, you'll talk about relatedness matrices. You calculate essentially the biallelic correlation at each locus for 2 individuals, and then sum across that individuals to get a basically relatedness measure. And you can compare spouses to the population mean, which would be 0, and we find that spouses in the United States with no tradition of first cousin marriage, like many countries in South Asia and the Middle East, for example, where over 2/3 of marriages are actually between first cousins, known first cousins. We still find that because of ethnic stratification, you know, intra-ethnic marriage, and because of assortative mating on observed phenotypes, that we're the equivalent of first cousins once removed. If you take out the ethnic effect, then we're equivalent of second cousins. So that usually freaks people out. But overall genomic similarity between spouses tells us little in terms of the genes that matter, so to speak, for the outcomes we care about. So we looked at the polygenic score, simply the sort of association between spouses' polygenic scores, and you could see the lighter bars are the phenotypes correlation between spouses, and the darker bars are the genotype correlation. And you can see, you know, educational attainment is correlated between spouses over 0.5. The polygenic score for educational attainment is correlated at 0.15, much less, but it's not trivial. Interestingly, other ones that are not observable, I mean, of course, height and education are largely completed before marriage. Body mass index, couples can grow fat together and affect each other. And depression, believe me, if your spouse is depressed, it's going to drag you down too. So, so there's intra marital socialization effects, and there's assortative mating, two different things going on here. But interestingly, we find zero correlation between these particularly noisy scores, though it should be said, for BMI and depression of spouses. I think this is the most interesting thing worth examining, the fact that for height, the genetic sorting is actually higher than the phenotypic sorting, which suggests that it's actually, and other evidence is that the polygenic score for height is predictive of other development, healthy development as well, that height is a proxy for that. Yeah?
How much of this can be explained by the fact that the polygenic score for height explains a significantly larger proportion of the variance?
It should not be explained by that. There is, we did a number of things like correcting for measurement error, it's not driven by that. So as you know, if it's on the measurement errors on the right-hand side, it's a problem. If it's on the left-hand side, it just makes the standard errors bigger. But when we've done it with phenotype, genotype correlation, and then we've switched, so we can measure the phenotype without much error. We can switch them on the side, so one's on the right-hand side. We can do something called simulation of measurement error, where we add measurement error, and then we plot what that does to the in terms of attenuation bias to the coefficient, and then you can plot that curve, and then you can back it out to continue the curve under some assumptions to what it would be if it was zero measurement error. So all of that suggests that this is not driven, the differences you're seeing between these are not driven by measurement error. That answers that. So we asked though whether or not this is changing over time, and the answer was, this is for phenotypes, yes, there's more education stratification today, or sorting today than there was 50 years ago. But when you look at the phenotype, the genotype, there's nothing going on for any, so we're not increasingly diverging within the US population. Okay, I'm really out of time, right? So I'm gonna skip all of this. But fertility, sensitivity, we're really well powered for height and BMI. We're not as powered for depression and education, but we're not terrible. I won't go through all of this. Okay, so this was the example I gave, gene for aggression in South Bronx. In the 1970s, when I grew up, you end up, I don't know, in jail. This is Mitt Romney's house. You end up as CEO of Bain Capital if you have the same gene for aggression. But how do we estimate that? This was the early famous paper by Avshalom Caspy et al. That claimed to find a gene by environment interaction for a single locus, the serotonin transporter, where if there was no male treatment, there was no genetic effect of the difference between alleles, but if there's childhood maltreatment, you found this genetic effect. However, this is endogenous environment, which is an important take-home lesson. So the parents, untransmitted alleles, the parents, all the entire genome of the parents was also passed on, 50% of it to the kids, and they're probably the ones who were maltreating them. So we don't know if it's the maltreatment or the genes that were leading the parents to maltreat the kids that is in a gene by gene interaction with this locus. So that's all to say. Another example from sociology is family meals, add health again, and delinquency. Of course, though, that's social organization of the household is itself endogenous to the genotype of the parents and the kids, for that matter. So we really don't know if it's gene-gene or gene-environment. So you need an exogenous environmental shock. This is the dumbest politician in the world up until now, because he was sending kids off to Vietnam with the draft lottery. He's a congressman from upstate New York. I thought it was a good re-election strategy. I swear that must be Robert Moses, but I can't. Anyway, draft lottery, bad for 18-year-old men in 1969, but great for social scientists. Random assignment, we find in the tobacco, this is work with Lauren as well, that those people on the top end of the polygenic risk score distribution for becoming smokers are the ones that show an effect of being drafted in terms of their lifetime smoking behavior and therefore lung cancer risk, because they're exposed to a very tobaccophilic environment by being drafted into the military. They were given cigarettes, they were cheap, norm was to smoke. So people who had high polygenic risk for smoking demonstrated an effect, but those at the lower end of the polygenic score distribution did not. I don't have time for this, but we looked at the effect of PGSs within the family versus cross families, because we talked a lot about this morning. I'll take 1 minute to just say this, because I think I think the assumption of random distribution within a family or within offspring of the polygenic score or a particular allele is a pretty safe assumption. However, the internal validity, and so the internal validity of that estimation is strong, but the external validity is under question because, as I was talking about in the break with some people, you could imagine that using this ideal world where you do sibling fixed effects or control for parental genotype, particularly the sibling fixed effects, you're identifying off the difference in the sibling. So let's say you, Dan and David are both like, David's +2 standard deviations in his education polygenic score. David's only +1.8. But I'm, as a parent, have to make investment decisions, and I may drastically enhance that small trivial difference because I'm having to decide within the family who to allocate resources to, like reading extra books or sending to private school or whatever. So the question is whether or not we can generalize from these sibling models to population-based distribution of, say, the phenotype.
And the direction isn't obvious, right? It might be that you feel
right.
bad that Dan went to college and I don't get to.
Right, and there's also evidence that there's class differences in investment patterns, so that higher SES families are actually compensatory investors in their children, and lower SES families are reinforcing investors in their children's ability. So all these things become complicated when you want to think about it, and even more so, I think this is really interesting. If you look at siblings, so this is in the Minnesota twin family study among fraternal twins, and my polygenic score, 1 standard deviation increase in my polygenic score predicts, with controls for PCs, predicts a little more than half a year more schooling, okay? But conditional on my parents' genotype and my genotype, my siblings' polygenic score predicts as well. And then when you break this out by sex, what's really interesting is that it's driven by sisters. They only have same sex, unfortunately, pairs in this data set. But sisters have an effect. So if you're 2 sisters, not only your polygenic lottery matters, but also your sister's matters in a positive way. For brothers, brothers don't give a shit what their brother's genotype is. There's no effect. So this suggests that there's a lot more complexity to the within family dynamics of genotype, and there's possibly SUTVA violations and so forth. This is very suggestive early evidence that I'm working on and trying to replicate in other data sets, but something to think about as you go forward. That I showed the earlier slide where the effects controlling for PCs across families were the same as within families, but that might not be true for all phenotypes. So for example, Peabody picture vocabulary test, big differences between cross-family models, within family models, also for depression. So there are areas where genotype might lead to accentuated niche formation within families or cross-sibling effects that should be thought about and investigated. So I'll stop there.